In today's class, I will talk about amino acids and proteins, the building blocks of our macromolecules. So we will talk about amino acids and proteins. So as we know, amino acids, they are the building blocks for proteins in our body. So and they are similar, proteins are basically similar from from bacteria to humans. They are made of the same set of 20 standard amino acids. They are made up of the same 20 standard amino acids. Out of them, 19 of them are alpha amino acids. I will explain what is an alpha amino acid, and one is an amino acid. One is an amino acid. So if we look at the structure of an amino acid, you have a central carbon known as the alpha carbon, and to one is attached a hydrogen, to another is attached a carboxyl group COO minus, and another is attached an NH2 group or NH3 plus. You can call them as NH3 plus, and to this is attached an R group. This R group can be anything. Depending upon this particular R group, you have the character or nature or property of the amino acid, and eventually this influences the nature of the protein also. So this is an alpha amino acid, structure of alpha amino acid. Now according to if we talk about DL nomenclature, when the NH3 is on the left side, this is an L amino acid. But when the NH3 is on the right side, this is a D amino acid. So this is how the basic nomenclature of amino acid stands, how the DL nomenclature stands. In nature, physiologically, and living animals have L amino acids. So all the amino acids, there are few exceptions. All amino acids are basically L amino acids. কে ঢুকছে দাঁড়াও দেখি। Now let us look at what are the 20 standard amino acids. So as you have seen that the 20 standard amino acids, they have a similar structure. They differ only in the R group. Physicochemical nature of the amino acid is only determined by the R group. So on the basis of this R group, you can divide amino acids into the first group. Let us talk about the first group, which are the hydrophobic, aliphatic amino acids. They can be hydrophobic, aliphatic amino acids. So let us have a look at the structure of these amino acids. As per the amino acid structure, the simplest of them is glycine. So since all are L amino acid, COO minus H, and H is present over here. It is written as glycine. You can write it as GLY or single letter G. This is the simplest amino acid, glycine. Hydrogen is hydrophobic. Then the next amino acid is alanine. This R group changes. You have H, and here instead of H you have CH3. This is alanine. Now you remember one thing, that there are D amino acids and L amino acids. So all the 19 amino acids, and of course one amino acid is also there, also one amino, that is 18 plus 1 amino acid, proline. They are chiral molecules, and only glycine is achiral. So a chiral molecule is a molecule which is attached to 4 different groups. The central carbon, this central carbon, it is attached to 4. This is a chiral carbon. This is a chiral carbon. This is attached to 4 different groups. You have CH3 is one group, H is one group, COO minus is one group, NH3 plus is another, 4 different groups. In the case of glycine, it is achiral because it is these two groups are the same group. It attached to two hydrogens, so it is not. Glycine is achiral. So you don't have any L-glycine is not present. L-glycine, D-glycine, this does not occur. Such cases don't occur because it is achiral. So like glycine, you have as you see, you have the next valine, you have leucine, you have isoleucine, methionine, proline. So proline is an amino acid where whose structure is a bit different. H2N plus, remember, C the H is attached over here, and from here you have this CH2 comes out, and it is attached like this. So instead of an amino group, you have it's an amino group, amino acid. This is imine. Remember, proline is a strong alpha helix breaker. We will come to it. So you have cysteine and you have methionine. All these amino acids are present. The next group of amino acids which are present, the next group are the hydrophobic aromatic amino acids. The hydrophobic aromatic amino acids. Among them, the hero among them are phenylalanine, tyrosine and tryptophan. Similarly, you can see that the structure is same. You have H3N on one side, COO minus, and H is attached, and in the R group you have showing the resonance. This is the structure of phenylalanine or PHE. People call it F, not P, because proline has taken it away. The next one is tyrosine. It's the same thing, COO minus, CH. You have, and you have this ring group, and you have an OH over here. This is this, because of this OH, this is tyrosine. Tyrosine has got TYR, or it's written as Y. Similarly, you have tryptophan present over there, another amino acid. The next group is the polar charged. So far they were uncharged, polar charged amino acid. So let us look at first among the charged, the positively charged amino acids. Famous among them is arginine. Similarly, you write H3N, and here is the R group. You have a CH2, CH2. After 3 CH2 comes an NH. Then comes a C double bond NH2 plus, and there is another 1 2 3 4 NH2 plus. So basically what you basically have in this particular molecule, this and this, they balance each other, but there is a net positive, net +1 charge present. Because of this net positive charge, this is a basic amino acid. Similarly, arginine, you have lysine, you go in the same manner. Lysine has got just one more carbon more. CH2, CH2, चारटे CH2. Then you have NH3 plus. So this and this balance, and you also have a net positive charge. This is a basic amino acid. Like that, you have a, these are positively charged amino acids. Like that, you also have negatively charged amino acids. They are also these are known as basic amino acids. These are also known as acidic amino acids. Famous among them is aspartate and glutamate. Let's have a structure at aspartate. The same structure, the R group has got a CH2 and CO minus. This is the structure of aspartate, ASP or D. Glutamate has got just one more CH2 over here. So this is a, here what you find, this and this, they balance each other, but what you have is a net one negative charge. And they are polar, means one side plus, one side minus. They have got charge is present, therefore polar. That is, these are water soluble. They are water soluble. And the hydrophobic amino acids are not water soluble. Amino acids are not water soluble. So the next group which we will study are the polar uncharged amino acids. Famous among them is the serine. You draw the alpha carbon, the COO minus, the H, and the R group has got just another COO minus. Sorry. It's got a CH2OH, an alcohol group present. They are polarized. That is, this is alcohol, this is water soluble groups. These are water soluble groups. So alcohol is water soluble. So this is serine. Similarly, you have threonine, asparagine, glutamine present. So after this we go to the next concept, that is the concept of peptide bond formation. So let us draw this peptide, how the peptide bond works. So let's draw this amino acid H3N plus to this. I am drawing it a bit differently. The H is kept over here and I am drawing a COO minus over here, and this is the R group 1. First R group, R1 group, plus another amino acid. R2 now. So let's, what happens? The peptide bond formation takes place. You have a water molecule coming out. You have a CONH group formed. R1. CO and here NH. O minus comes, and so and from here a water molecule gets out. So this bond is known as CONH. This is the peptide bond. So the peptide bond has got a partial double bond nature. That is because of this CONH, this electron can migrate over here. This electron pair, it can migrate and resonate. So constantly this electron is coming here and it's going over here, it's resonating. Result is that it is neither it is stronger than a single bond and weaker than a double bond in between. This is weaker than a double bond. So these peptide units are, let us use a different color. The peptide units are rigid and planar. That is, let us draw this peptide bond properly. Let us draw it once more. We are drawing the first R1. Here is R2. Then we are drawing the next amino acid, CO, NH, CH, this is R3. Then you have CO. So this is how, when you look at this structure, you will see this region. This is one rigid planar peptide unit. So these are actually this region, they are rigid, they lie in a plane, there is no rotation over here, single plane, they lie in a single plane. This can rotate, this rotates, this rotates, this rotates, the CC rotates and CN rotates. This bond angle can rotate or this bond angle can rotate. The C alpha N, C alpha can rotate and C alpha C, they can rotate. But CONH, this does not rotate. This whole unit rotates as a plane, মানে এরম করে যাবে, এরম করে যাবে। So they move as a plane. CONH doesn't rotate. So let us have a look at these angles. The structure. Let's draw this properly. The structure properly. This is C alpha. A bond is going down, goes to a carbon. It is attached to an oxygen. This is attached to a nitrogen. This is attached to a hydrogen. The N is attached to another C alpha. This C alpha is attached to a hydrogen here, and it is attached to another hydrogen here. It is attached to another carbon which is attached to a oxygen. This C is attached to a nitrogen. This is attached to another C alpha. This is attached to a H. And now what the plane is, this is one plane, and this is another plane. So this angle psi, phi, and this angle over here, C alpha C, the NC alpha, this is this phi bond, and this rotation over here, that is C alpha C, this is known as psi. So these angles are psi. So for different amino acids have different R groups. They have different R groups. Different amino acids have different R groups. This is an R group over here. Let's different R groups. So on the basis of this R group, this R group and say this is attached to another R group over here, CONH. This is attached to say another R group. If they are similar charged, then they will repel each other. If both are, one is hydrophobic and one is hydrophilic, they will repel each other, and so these angles will be different. And on the basis of these angles, this psi and these theta angles in a polypeptide chain, a linear series of amino acids. Based on these angles, essentially what you have, the structure of the protein depends. This is one is 90-degree, the next bond moves at it moves at this angle. The next one again takes this angle. Next one is even more angular. So like this, the shape goes on like this. So here it is straight, then it takes this angle, then it takes this angle, and then it goes even more steeper like this. So something like this. It's even steeper. So this is how the structure of an amino acid takes place. So now we go to what is primary level structure of an amino acid. Primary So the primary structure, primary level of structure of a protein, primary structure of a protein or polypeptide, primary structure of is linear sequence of amino acid as joined together by polypeptide. It is the linear sequence of amino acids as joined together by polypeptide bonds. So this is the sequence of nucleotides determine this structure. Sequence of amino acids in a polypeptide. So this is how, this is the primary structure. Inside primary structure also is included disulfide bonds. So let us see, actually disulfide bonds, they form where there is cysteine residues. So let us look at a cysteine molecule. We are drawing it a bit differently. CH2 SH. This is one cysteine molecule. We are drawing another cysteine molecule to this. This is another molecule. They are in a reduced form. Under oxidative stress or under when oxidation takes place, what happens? Two hydrogen comes out. That is, they move away like this, and result is that this hydrogen, this has joint like this. So it has become like this. This is a disulfide bond. So this is a polypeptide chain. For example, there is an SH group over here, another polypeptide chain, they have got an SH group over here. Or for example, you have one molecule, it has got an SH group over here, another molecule SH group over here. Then a disulfide bond, if it forms, like in the case of antibody, this is a disulfide bond which takes place. And essentially what it does, it folds this molecule. So the polypeptide gets folded because of the disulfide bond. Now we go to the secondary structure of protein, secondary level of structure of protein, or however you call it. So the secondary level of structure in a protein is the regular folding of regions in the polypeptide chain. So because regular folding, because of the bond angle, the psi and the phi bonds which you had seen, because say R1 and R2, their particular relationship with each other, if they are similar charge, they will repel each other. If they are hydrophobic, then they will pull each other. So based on this, the protein structure also starts to fold because of the turns and twists because of these R group. So the secondary level of structure in a protein, in a protein is the regular folding of regions, is the regular folding of regions of the polypeptide. So basically their most common types are the alpha helix and beta pleated sheet. Apart from that, there is a beta turn or beta bend people call. So what is alpha helix? Basically, responsible person, the responsible factor is as the presence of hydrogen bonds. So in an alpha helix, the structure goes, this is the C alpha. This is the C alpha of the consecutive. Another C alpha. Next C alpha. So this is basically, the structure is something like this. So essentially this much is 0.15 nanometer. From here to here it is 0.54 nanometer. .6 amino acids per turn. So this is how basically an alpha helix looks like. So the folding is because, let's draw the structure once more cleanly. Why do they turn so much? So let's draw. NH, CH, here you have R1, CO, NH, CH, you have R2, CO, NH, CH, you have R3, CO, NH, CH, R4, CO. I am just drawing one after the other. NH, you have CH, say R5 over here. And now say from this oxygen and this hydrogen, there is a hydrogen bonding attraction. This results in 3.6 turn. 1 2 3, you see 1.2, approximately 3.6 amino acids go per turn because of this hydrogen bonding, and that this is determined by the nature of this R group. So there are specific amino acids which favor alpha helix. There are specific amino acids which prefer beta turn. So let's go to beta pleated sheets. Beta pleated sheets. So we are drawing this again. CH on top, R, N, H, then the C double bond O, then again CH, you have an R group, then NH, then again the CO, then again the CH, you have the R group again here, then you have the NH, you have CO, CH, R, again NH. This basically goes on and on, repeats. And now there is a hydrogen bonding with, say, an oxygen over here. Hydrogen bonding with a hydrogen over here. Again an oxygen hydrogen bonding. A hydrogen stays over here. So we draw the structure as, it's very simple now to draw. You people can remember it very easily. CH, after CH comes this CHR. Immediately after CHR comes CO, then this NH, CONH group, then immediately after that comes CHR, then again the CO comes, NH, CHR, NH. So this is a beta sheet. You can see it's again the player over here is, in both of these cases, it is the hydrogen bond which holds. It can go like this, then there is a turn, so you can see that it is the hydrogen bonds in between, say for example, which holds this beta pleated sheet together. Similarly, even in beta bend, when this turning takes place, there is also a hydrogen bonding which takes place in this region. There is a beta bend bonding which takes place. So this is how the alpha helix and beta sheets are there. Now we go to the next level of protein structure, and that is the tertiary structure. So the third level of structure found in protein, it refers to the spatial arrangement of amino acids that are far apart in the linear sequence, as well as those residues that are adjacent. So let us have a look at a particular structure. For example, if some amino acids are hydrophobic, what will happen is they will try to go to center away from water. So if this is an amino acid, a polypeptide chain, and you have these amino acids, say for example, if these amino acid residues are hydrophobic, so what happens when this protein will fold, you will always find the hydrophobic residues always will go to the center. As usual, this is also determined by the sequence of amino acids. Because of these are hydrophobic, they will go to the center, and if these are hydrophilic, these regions they are hydrophilic, just hold on, I have just done a mistake over here. Mistake is that I have pressed a button on my pen, and that has, so if these are hydrophilic, they will stay towards the outside. So this is the tertiary structure. Apart from that, you have the Next level, quaternary structure. This is, for example, in hemoglobin, you have one polypeptide and another polypeptide. They come together, they associate. For example, in hemoglobin, alpha 2, beta 2. So alpha polypeptide and beta poly 2 alpha, they associate, and these two polypeptide chains together, and they come and they form the quaternary structure of the protein. So with this I will stop my sharing today, and this ends my class today. Do you people have got any questions? Please feel free to ask. Questions আছে? হ্যালো,
হ্যাঁ স্যার।
হ্যাঁ বলো, জিজ্ঞেস করো, কোয়েশ্চেন থাকলে জিজ্ঞেস করো।
স্যার বিটা বেন্ডস এর জায়গাটা আরেকবার বলুন।
বিটা বেন্ড,
হ্যাঁ
বিটা
স্যার।
বেন্ডটা কিছুই না, ওই যে যখন বেঁকছে, so let us talk about beta bend. Just hold on, it is not getting shared for some reason. So let us draw the structure of a beta bend properly. You draw C O, মানে যেখানে বেঁকছে আর কি, CH CHR, NH, আমি ব্যাকাচ্ছি এখন, বেন্ড, I am trying to twist and turn them. CO CH R, after that will come an NH, NH, CO, CHR, NH, CO, CH R, NH. Now this, for example, there is a hydrogen bond over here. So because of these R groups, this is bending. These hydrogen bonds are holding the beta bend properly. So this is when it's twisting, say it's going in this direction. So this is a beta bend, this is in a beta pleated sheet, it's going in the other direction, so this is a beta bend. So this is how it goes. Anybody has got any questions? Any more questions? Please ask me.
স্যার ওই ইমিনো যেটা বললেন না, মানে বাকিগুলো একটাই খালি ইমিনো আছে,
হ্যাঁ,
স্যার ওটা
প্রোলিন।
শুধু প্রোলিনটা না ভ্যালিন প্রোলিন
না
দুটোই
না,
হবে?
only
শুধু
proline,
প্রোলিনটা তো?
only proline.
আচ্ছা,
I have drawn the structure of
বেশ।
proline, it's only proline. Proline is an amino acid, others are all amino acids. And
ওকে স্যার।
proline will break any alpha helix anywhere. If an alpha helix of a protein comes, the moment a proline comes, that alpha helix is broken. It's a very strong helix breaker. Proline. Any more questions?
স্যার,
হ্যাঁ,
স্যার বলছি এই হাইড্রোফোবিক রেসিডিউ গুলো যে কয়েল হবে থাকে চেনের মধ্যে।
hydrophobic. Hydrophobic residues will always try to go in the center. They will go get away from water. তাই জন্যে, for example, in a winter, if somebody throws water on you, what do? You sort of shrink yourself inside. গা গুটিয়ে বসে পড়ো ওরকম করে, so you are hydrophobic at that time. So these hydrophobic
আর
residues, they, they will always come towards the center. They'll remove, the water will be on the outside. For example, whenever it's almost whenever there is a hydrophobic any hydrophobic thing, it hates water, it will go away from water. This is this is about so it doesn't depend on whether it is a helix or whether it's a, there are certain amino acids which are helix forming, there are certain amino acids which are beta pleat, they form beta pleat sheets. But hydrophobic, hydrophilic, it is a different, it gives a different level of structure. That is the you are getting the tertiary structure. The secondary structure is alpha helix, beta. After alpha helix, say for example, if I draw here, এদিকে alpha helix is there, then suddenly it starts, I'll use a different color. A beta bend starts. If there are amino acid, if this beta bend structure, this region, this beta bend, this has got hydrophobic regions. So if it goes, this is the alpha helix is going on and on and on and on. So suddenly there are, this beta bend is there, the beta bend region will be inside, it will try to get inside, and then again say alpha helix comes, it comes outside, then suddenly there is an alpha helix region which is hydrophobic, it will go inside. So irrespective whether it is an alpha helix or beta bend or whatever it is, it will go to the center. If it's a hydrophobic or hydrophilic, hydrophilic will be outside and hydrophobic amino acids will go into the center. It is like your whole face. The whole face will go inside. When you're feeling cold, you put your face inside the blanket. So your face has got your alpha and beta helix configuration, but it goes towards inside under the blanket during the winter. So this is how actually you must look at it. This is the next level. Alpha helix is the second level, and hydrophobic, hydrophilic, which you are talking about the third level. First level is only the linear, the R groups. Any more questions? কারো কোয়েশ্চেন নেই? তাহলে আজকে এখানেই শেষ করি। তোমাদের রেকর্ডিংটা পাঠিয়ে দিই ইউটিউবে করে, লিংকটা
স্যার
পাঠিয়ে
নেক্সট ক্লাস কবে হবে?
আমি বলে দেবো। ওকে ওকে, আই এম এন্ডিং মাই শেয়ারিং রাইট নাও। I'll send you the recording. Next class I will go about with carbohydrate structures. Okay, bye. If you have questions, you can write to me.